My name is Kwabena Baduin-Kansa. I'm a lab technician in the Heinz lab here in the Koch Institute. So what we work on are cell adhesion molecules. These are molecules that line the exterior of the cell, and they allow the cell to sense things in the environment outside of them and translate those signals to the environment inside. One of those signals induces contraction, and so that's one of the things that we're interested in now. One way of actually looking at that is we isolate a cell line called smooth muscle cells, and these are cells that wrap around large vasculature. And you can imagine that they experience pulsatile forces because of the heart. The heart pumps fluid in, and you have to have some sort of property of the vessel that keeps it from essentially, dilating, maybe exploding in some cases. And that's one of the things these cells do. And they do this by contracting in a way that applies a force that opposes the pulsatile forces inside the blood. Normally cells are grown on plastic, but in order to get an idea of how cells respond to something like stretch or contraction, we use the silicon membrane, which is very elastic. So when we pull it, we can see how the cells on this membrane respond to the fact that the thing they're sitting on is being stretched. And so in this picture, that's what you kind of see. You see the cells responding to contraction. They elongate and they align with each other in one direction or the other, depending on how we induce the contraction. What we've stained for in these cells is a protein called actin. And the actin essentially makes up the skeleton of the cell. And you'll see that when you induce contraction, that this actin gets remodeled. And in this case, we've stretched the membrane, and you can see that there are large actin filaments that are kind of aligned, and that tells us that the cells can respond to this contractile force. When we take away these cell adhesion molecules, the molecules that surround the cell and transmit these signals, you won't see these actin cytoskeleton also get bigger or smaller. You won't see this remodeling event take place. This is also important in cancer, for instance, because as the tumor cells become more aggressive, they can hijack this response, and that allows them to invade the tissue around them and actually move in and out of vasculature. And so you can imagine if we were to figure out what proponents of this contraction response is important, these could provide clues on how you can target these tumor cells and help stop them from moving out and essentially invading the space around it.